As was just said, I'm using a biomolecular approach to the study of pigeons dispersal, and my samples come from the Roman to medieval period in Oxfordshire and then also in the UK. So, first, I just have some questions for today that I want to kind of direct where we're going. First, what is bioarchaeology? I know that we have some archaeologists, a lot of archaeologists here, but I don't know what level or what regions you guys focus in. So I just want to make sure very briefly that we're all on the same page of what is encompassed by bioarchaeology when I'm saying we're using bioarchaeological approaches. The second question is, what is a pigeon? Seems like a really basic question, but since I met Shreya and Simran, I've had more meetings with my primary and secondary supervisor, and we've gotten in these conversations where we don't actually know what falls under what is a pigeon. So we've had to do a lot of work and research to really refine our understanding of what actually is a pigeon, because there's so many different species and variations and whatnot. And then we're going to get more into the archaeology of what is the history of the pigeon, because while we may just think of those sky rats on the street, they have a really long, complex history with us, and we're really integral for thousands of years to our survival and companionship. And then the fourth question is, what actually is my project? So we're going to look at that. The fifth question is a bit of a lie because I don't have the answers yet, but it's the main question of my dissertation, which is, what is the dispersal history of the pigeon? And then we're going to finish off with some questions from you guys. So first, to start us off, what is bioarchaeology? So bioarchaeology is a study of anything biological or organic, which is really broad and really awesome, in my opinion. There's three main things that fall under bioarchaeology when you're doing any kind of analysis in that way, and that's going to be lipids or fats, proteins, and DNA. So lipids are going to be medium-sized biomolecules. They're often, but not always, extracted from ceramics, just because that ceramic matrix really stores onto those lipids quite well and preserves them and they're the most abundant. So it's just commonly done on ceramics. And we can reconstruct past human activity and diet through compound-specific dating, analysis, lipid analysis, wherein you can understand what types of dairying fats were being used. Was it for ruminant fats, adipose fats, meaning was it from multiple stomach things like a cow, single stomachs like a pig, like horses, us. And then we can look at proteins, which are typically the largest of the macromolecules, and they then hence preserve the best. So you often can get proteins extracted from the oldest material, typically much older than DNA, unless there's extenuating preservation circumstances. They're made up of amino acids, and they can be species specific, and there's many applications and uses. I used to work in Zoom, so I'm quite passionate about proteins. I think they're pretty awesome too. Proteomics is really, really cool stuff. Same with dental calculus and looking at macrofossils preserved in teeth. And then finally, I think what I have to say is my favorite, because I am doing it, is DNA. So it's the smallest and most prone to damage, but it's also often the most informative. So DNA stores, dictates the information that makes up an individual, their genetic history, diseases, and inherited traits. But something to remember with DNA, I was watching this really awesome talk by Beth Shapiro, if anyone else is familiar with her work, she's kind of awesome. And in it, she's talking about, imagine you're at a parade and confetti goes off. All those little tiny different colored pieces, they explode all over the street. Then it rains all night, winds come through, people are all walking over them. In the morning when you come and there's these torn up little disgusting shreds of paper in different colors and whatnot, think of that, of what the preservation state of DNA is that then we're working with when we're looking at ancient DNA. So it can be quite tricky to reconstruct and understand what we've got going on. So I want us to stay focused on what is the dispersal history of the rock dove slash feral pigeon. We're going to get into why feral pigeon is quite a problematic term, but is commonly used throughout all the literature. But just to remember, this is the main question, even though I don't have any of the answers currently. And then first, we're going to start with, what is a pigeon? As Shreya has said, I love to take pictures of pigeons now. So that's some nice pigeons in London enjoying a bagel that entertained me entirely too much. And I thought maybe you guys would like it too. So the first thing that we start with not only what is a pigeon, but what can we call a pigeon? And what me and my team has decided we're going to use is the anthropidge or anthropigeon. because of some really complicated things that we need to remember when in a lot of the literature, they refer to a pigeon as being a feral pigeon or a domestic pigeon. So first of all, if we're looking at this wonderful graph that my supervisor was actually a part of a paper on, if we look at the different stages in domestication, because that's what this paper is aiming to explain, is a universal applicable definition for domestication, because it's so much more complex than we like to think of it as, is that we start off with these things called an obligate recluse. So they're going to thrive in the absence of human disturbance. So if we're not there, they're doing great. They're really happy. So we've got things like wolves, bobcats, different stag beetles, different insects, bacteria, such like that. We then move on to human avoiders. So these are things that thrive in areas where human disturbance is rare. So some species of pigeon will fall under that, but definitely not our anthropidge. We then have human tolerant. So those are ones that, they don't mind us. It's okay. They would kind of prefer if we weren't there, but it's okay if we're there. A human exploiter, they're going to thrive in these areas with ongoing human disturbance. So those are different species of flies. If you think of currently, especially, I don't know about you right now, but if I even leave my window open for a second, I have so many little flies in my kitchen. Carps really thrive with ongoing human disturbance. And then when we look at obligate synanthrope, those are the kind of things that are dependent essentially on us to retain their populations. So it's going to be things like maize, because we've made them so dependent on us in order to just reproduce. But you can also then see that different variations of maize fall under different stages of that domestication kind of linear timeline here. Well, not a timeline, that was a misspeak, my apologies. But if you can just see that teosinte, that's the original species of maize. So then we're moving through of it's more and more dependent on us. We're going to get the same thing that I think makes it more understandable if we look at dogs. So dogs depend on us now. They need us to feed them for companionship. I mean, I think a lot of people probably have a dog, have a pet. Same with the cat. They're going to fall under that same kind of designation. But if we then look, we have this thing that can also happen with dogs called feralization. So they're going to look the exact same because morphologically they have those same genetic impacts that occurs with domestication, but they don't have the same temperament. So they have been feralized in that you couldn't just go up and pet them on the street like you would with someone's pet dog. And the same thing happens, so when we then say a feral pigeon, that implies that they are like rabid or feral or going to attack us, but that's not what's going on. But they're also not domestic because we're not dependently feeding them. They're existing on their own. So we have things that are the rock dove, which is the progenitor of the pigeon. And then we now have this domestic / feral / now preferred anthropidge is the terminology to understand because it's anthropogenically, so it's modified by humans, pigeon. So I know that was a lot. I hope we're all still up to speed, if that makes sense. Hopefully I explained myself well there. So now looking at the UK specifically, because that's where all my samples are from, there are 6 main species of pigeon that we're going to be concerned with. And those are, as I just mentioned, the rock dove, which is the progenitor of almost all pigeon species, including the anthropogenic pigeon, things like the Danish tumbler, all these different kind of fancy pigeon breeds that we get. They all come from the rock dove. We also then have something lovely called the stock dove. We have turtle doves. Collared doves are present in the UK, but regarding my research, we're not going to worry about them because they don't come to the UK until after 1950. So while if you come to visit, you'll see them all over, they're not something that I'm going to concern myself with. And then the lovely wood pigeon. So again, the wood pigeon is something that's not of concern personally for my research, because if we look at this lovely table by Dale Sargentson on the left, and if we look at the weights, we can see that the wood pigeon is almost a third larger, if not larger than a third, than the rock dove, feral pigeon, and the stock dove. So it's significantly larger. So when we're looking at things like zooarchaeological assemblages, you can identify a wood pigeon versus those 3. However, when we look at things like the stock dove and the rock dove and the anthro pigeon, they are indistinguishable from each other. So some zooarchaeologists will claim that they can differentiate them, or as my collaborator at Exeter, the lovely zooarchaeologist Sean Doherty, he has said, he's like, I don't know how people are saying they can do that. There's morphologically like nothing that distinguishes them concretely enough that you can tell the difference. So in a lot of previous literature, how they also differentiated them was looking at the known dispersal, like the known, migratory patterns of the stock dove and the rock dove, where if pigeon bones were found up in the north of the UK, they would then designate them as rock doves because it was known that the stock dove didn't go that far up north. Now they're all ubiquitous, almost everywhere, all over the UK. But in the past, that was how they did it. I'm not saying I agree with that, but it is how it was done. The turtle dove, also very lovely and small. You've probably heard of turtle doves in various Christmas songs or seen them in religious texts and whatnot. Um, so they could be a small issue in regarding my research of being within the assemblages, but they definitely weren't eaten to the same way because of those strong religious associations with them. And another thing that has to be considered zooarchaeologically is that, spoiler, my pigeons all come from dovecots, which don't worry, we're going to go over what those are. But there can be juvenile birds, so that can make the sizes different. So essentially, there's lots of things to consider, but those are the UK's big six. So those are the pigeon species that I'm going to be worried about when it comes to my research. But just kind of go over pigeon reproduction. I have learned a lot about pigeons recently, and so are you. You're about to learn. So pigeons are monogamous, and their breeding cycle begins with first the courting phase, which is seen when maybe you've heard of those when they're cooing really loudly and doing the dance for each other. This lasts approximately 10 days, and within this period, the male and female will begin to construct a nest together. And then after about 10 days, the female will lay the first egg, and then approximately 24 to 48 hours later, she'll lay the second egg. So in a study in 1986, Holmberg and Clint, they took a bunch of anthro pigeons and in captivity and attempted to manipulate their breeding cycles to see what was the maximum amount of cycles that you could have year-round if pigeons were constantly being given food, because that's often what deters them from continuing to breed. And they found that within a year, with these 50 to 60-day cycles, they could get maximum 5 breeding cycles in the year. They also did some studies of seeing if they removed one of the eggs, if the female pigeon would lay a third egg, and found that while it did happen, it wasn't reliable, and it actually didn't increase the reproductive fitness of the babies at all. It often led to one of them then not receiving enough food later on. So this 17-day incubation period when we have the 2 eggs is the eggs are kept warm by both the male and female. They take turns. And then after they've hatched, this is, I think, the coolest fact I have learned recently about pigeons, is pigeons make pigeon milk. So it's in their neck area in what's called their crop. And they produce this really protein-rich substance, which is why, as a species, they are so reproductively successful in that there is just so many pigeons because they're able to get their squab or their babies up to weight so quickly in that week of feeding them essentially pigeon milk or crop milk. During this period, the male and female also both alternate keeping the babies warm for around 30 days post-hatching. And then after they leave the nest, they are booted right out. And kind of, I picture this like when you first have moved into undergrad and your parents kind of continue to drop in with like small bags of food to make sure you're eating. The male for a couple days will provide food for the squab as they're on the ground around where the nest would have been, just to support them in that first integration period. So if we then think about that's how reproduction happens, what about plumage and coat color in domestic pigeons? So when we think about pigeons, we need to remember that there is the wild rock dove, which is the progenitor, which we're going to go into more. But then we have this anthro pigeon, which I'm sure as you've seen, as the image on the right shows, there's many different coat colors and changes and things that we can see represented in their coat color. But within this, they have this wild type morph. So that's what we're seeing in the image on the top left. Number A is the wild type morph that then can make it really difficult for biologists who want to monitor rock dove colonies of pure rock doves, because you can have these anthropigeons integrated in, and it can be more difficult to separate them out. So we get a wide variety of pigeons and of different pigeon coat colors, and I think also then leads to a lot of this discrepancy over pigeons and doves. As the two are synonymous, they mean the exact same thing. The original is the rock dove, and we then start to refer to them as a pigeon. My theory and Sean's theory being that as we proceed to view them in less positive a light, so too does the terminology that we use for them. But genetically, they're the exact same thing. So then when we go into studies that are trying to separate out these anthropogenic and wild pigeons for conservation purposes and also just for understanding genetically what's happened. So there's a really cool study by William Smith, wherein he did both DNA work on modern pigeons as well as live capture and field survey in order to understand the amount of feral integration that was happening in British and Irish rock doves. So that's because in the Outer Hebrides, so in Scotland and in parts of Ireland, that's where they believe the only pure colonies of rock doves exist. So he came up with, through his research, that there's three main ways to actually morphologically differentiate between the anthropogenic and the wild pigeons, and that's through differences in their cere shape. So the cere is the little part on the top of their nose, the beak, which you can see on the top right where it says swollen cere there. They have a slightly different head shape where the angle is just a little bit different. The size of their orbital ring can differ, but essentially these are really minute differences where unless you're an expert, you're not going to be able to tell the difference. No questions on that. That is not my expertise. I am not a biologist, but I do think it's really important to understand how the impacts of just the spread of pigeons all over the world can have the impacts on these still wild and endangered species now that they originated from. But it's all because of how they're still able to embody this wild morph type. Hopefully everyone's still with me. Sorry, I know this is a lot of science to start us off. And it's going to go a little bit more. I'm going to hold your hands through it. We're going to look at some ancient DNA here. Or sorry, not ancient, modern DNA that they did. But it's really quite interesting for my own future study. So here, when they collected feathers all over the UK, Scotland, and Ireland, trying to understand both the introgression and the dispersal history with the modern pigeons. And what they found, so they used three main study groups, and it was these captive, in air quotes, rock doves that were coming from, it was a collection in the Netherlands where they were claimed to be pure rock doves from a previous museum site. So they were dead, if that wasn't clear. And they had on record, though, that hybridization had likely occurred, as in they weren't being kept as pure, in air quotes, it sounds a little eugenicsy, as pure as they were believed to be, which is on the right in the box plot that we can see here with all those goldish yellow dots are those captive rock doves. So clearly we can see that they were not pure rock doves. And then the England and Isle of Man, Highlands and Orkney, Inner Hebrides and Arran, Cape Clear Island, Outer Hebrides, and Shetland, those that are all plotting on the left for the majority are the ones where we're going from these colonies where it's believed that you still have the pure colonies of rock doves. They then took known data from USA domestic and feral pigeons, so that would be both the street pigeon or anthropigeons and fancy breeds, and they looked at their DNA as well, and really interesting found that there was almost a complete separation, as you can see in that plot, between the wild rock doves and the feral or domestic breeds. And that led to them to have some really interesting conclusions just about what that meant about the dispersal history and the spread. And they called for ancient DNA studies because they wanted to understand if the rock doves in Scotland were actually just a really ancient feral lineage that had become separated. So that's where it's interesting for my project, as hopefully we'll get to see some answers when we start looking at the DNA. And one last, I know it's very scary. I know it's scary. So this is looking at the DNA admixture with all of the supposed pure colonies, and what their conclusions from this was, just what our takeaway is going to be, is that if we look at the mix between green and purple and yellow, where it's mostly green, that's where we're seeing the purest concentration of rock dove without hybridization from these domestic, feral, anthropogenic, whatever we want to call them, these human modified pigeons. So you can see in the USA domestic and feral pigeons in the top left, there is the least amount of rock dove, in air quote DNA there, but in the Outer Hebrides, it's almost the purest here. So that's what led to them to have some really interesting conclusions just about what this meant about dispersal and little population bottlenecks and whatnot. So that's enough DNA for now. We're going to look at one final phylogenetic tree, as this lovely paper is actually what's providing all the modern data for my study. So it's interesting just to consider the work that's been done on it already from the lovely Hernandez Alonso. So here he took museum samples from all over Europe, Asia, Africa, or Northern Africa, sorry. And he sequenced their DNA and then plotted them out to see what kind of clade history we could see. He mapped them on the right. We have what's called an outgroup. So it's where you use pigeon species that are not genetically related to what we're looking at. And we can tell that because of the name. So Columba guinea, Columba rupestis, they're related, but they are not the same species because we're looking at all Columba livia. So all of these would fall under Columba livia, and then with their own species differentiation as an extra Latin term on there. And they just saw some interesting, I don't know if you can see my cursor, interesting clade groups, clade being that they are the most genetically similar, and we can see that because of what's called a bootstrap value. So that here is the support values for these just different genetic groupings. I'm not going to keep us on here too long, but just for maybe the few people that can follow. I know it takes a while to get up to speed with these kind of tables. So moving on to some more of the fun history of pigeons. So when we think of pigeons, what I think a lot of people don't realize is actually the amount of research and use of them as what's called a model organism for domestication. So Charles Darwin, I'm assuming everyone's familiar with that name. He wrote on the origin of species, which was changed our whole understanding of evolution and history by looking at natural selection. However, what people don't often realize is that he starts his book off with a chapter on pigeons about artificial selection. So he himself had a dovecot where he kept different breeds of pigeons, and he studied them for their remarkable phenotypic variation. So that's what we mean by phenotypic variation. That's all the different ways that we can see on the right here that they can just appear differently. So there on the bottom left is what's going to look like our rock dove. So that's that wild type plumage. I can't remember if this one actually is just a wild rock dove or if it is a domestic pigeon or an anthropidge. And then we can start to see all those different variations. There's around 1,500 species of pigeon currently with fancy breeds that have been genetically modified just to have this much differentiation, where you can have pigeons without beaks, where they have to be hand-fed every day. You can have pigeons with furry, fluffy feet all over, with huge, big crests like this one does. It's just really quite remarkable, the amount of variation that you can have within them. And Charles Darwin recognized that, and he was the one that made the first conjecture that he thought that all pigeon species originated with the rock dove. And then this paper came along in 2013 and proved him right. So they sequenced a significant amount of fancy pigeons, which we can see some more of these fancy pigeon breeds here. I've put their names down if you're interested to know what species they actually are. But in sequencing all of these genomes, they then did confirm and see that they were all originating with the rock dove, which is really interesting that you can have this much phenotypic variation, but all coming from the same common ancestor. Sorry. So I think that's enough science for everyone. Hopefully you're all still with me. We're going to get into what I would probably say is a little bit more of the interesting stuff because it's quite fun and archaeological, and that's what is the history of the pigeon. So first, to start off, just make sure we're all on the same page timeline wise. This is the history period of the pigeons that the periods that my samples come from. So we're starting in the Roman period with, I've got a few pigeons from the Roman period, and then the bulk of my samples are coming from the early to post-medieval period. There is supposedly one Anglo-Saxon one. I just got my dates back. I haven't confirmed if there actually is an Anglo-Saxon one, but included in there just so we'd have that time period in there as well. So when we first start off with the pigeon, I'm actually going to start us off 67,000 years ago. So in Gorham's Cave, which is going to be on the Gibraltar Peninsula, we have evidence of cut-marked bones spanning a period of 40,000 years in this occupation history where Neanderthals were eating rock doves. So I'm definitely not saying they were domestic. That is not at all what's being said. But I think it's really, really interesting to consider that our relationship with the pigeon started long before we were even around. There was around 2,000 pigeon bones, or rock dove bones, sorry, that they were looking at. And as we can see on the left, the very clear cut marks are evidence showing that there was mass consumption of these pigeons over a really long period of time. So moving on to some more fun stuff, I've chosen objects that are all just from the UK. I just think it's more fun to have ones that are local to where I'm currently living, at least. So when we think of potential domestication for the pigeon, even the timing on that is highly contentious. Some trace its origins to the Neolithic period, where accumulations of grain around settlements created a niche for the species, and that is believed to have led pigeons to become one of the first domesticated birds. Others attribute its spread to the Romans, who, like with other exotic animals, may have transported pigeons around the empire, both as a food source, but also as a symbol of status. So if you're starting to get if you just have your own personal pigeon, that's a status symbol. Same with any other those animals that we see that they've brought around. And so if we look here at Tel Arpakiak in Iraq, dove figurines were found, which led to people to argue for a domestication age of 4500 BCE for pigeons. They're also seen frequently in Assyrian reliefs, and the ancient Egyptians even kept pigeons as large flocks as offerings for ceremonies. So, according to the Papyrus Harris, the offerings for Ramses III, which we're looking at, is around 1186 to 1155 BCE. They were given to the temple at Karnak, and it included 6,500 pigeons. But that's a lot. That's a significant amount of infrastructure dedicated to keeping pigeons just for religious ceremony. And it's demonstrating that pigeons have clearly played a part for a long time in the history of humankind, likely since the first agricultural communities. So here on the left, you can see a truly lovely bronze lamp that's actually at the Ashmolean Museum. I was entirely too excited when I saw it, when I just happened to be walking around. And it was found in Nubia. And then on the right is another oil lamp, and that's in the shape also of a dove flash pigeon, and it's in the collections of the Victoria and Albert Museum. It's coming from a lot later in time, but still really quite old. If we then move on, when we think about when we're looking at ancient Greece, we've got doves or pigeons, and they're often associated with deities. So for the Assyrians, it's with Inanna, and then here we've got the dove or pigeon being associated with Aphrodite, and then with Eros, who we can see here with his wings as he's holding a dove or a pigeon. We've got this beautiful gold finger ring that's depicting a pigeon on the front, and then Eros is on the back. Unfortunately, there wasn't a photo of him there. So these are both coming from the 5th century BCE, and the pigeons' association with these deities may have also motivated both their capture and dispersal, with these associations with Ishtar, to the Assyrians. We've got the Holy Spirit in Christianity, and then Aphrodite and Eros to the Greeks. So this long-standing association of pigeons for religious, however you want to take that word to mean, iconography, is really quite lovely. And then when we think of the Romans and this dispersal all around the empire, we have to think about some of our most beautiful preserved wall friezes. So this is from the House of the Doves in Pompeii, where we can see it's dedicated entirely to pigeons or doves. I mean, those look quite recognizable to me. It looks like some of the ones that I see on the street when I walk around and go to work. So we then need to start thinking about how are pigeons actually being housed. So when we look here, we can see a columbarium or a dovecot, columbarium being the word used in the Middle East and in Rome. For what we can see here is these step structures with pigeonholes that are put in, and that would be to keep them safe, keep them provided food, and also concentrate them so that you're able to get out both the really valuable resource being their iguana or their poop, as well as for meat. So this one's from the 1st century BCE, and it's supposedly a scene depicting on the Nile of a columbarium, but it was found near Rome. We can then start to look at some Iron Age dovecots, just so we get an idea of the range of how they can vary in their appearance, but also their similarities. And that we're always going to have these kind of cutout holes for them to roost in, as pigeons are so successful as a domesticate because of their tolerance for being in confined spaces. They really don't mind being walled right in and kept close. So these ones are coming from Northeast Oman, and the studies there if you wanted to read more about them. Looking at some examples for in the UK, so we've got a dovecot here from Avebury in Wiltshire, and then on the right, a dovecot from Wales. So just we can see they're being made both in clay, as we can see here, and then from stone over in the UK. So you're using what resources you have, but the general construction is relatively similar. Now, when we start to move on, we want to start thinking about the medieval period. This is a really lovely painting from the Ashmolean. Unfortunately, it doesn't show the pigeons, but they would have still been present. So we have what's called these royal forests, or for the medieval hunts, wherein you're going to be using your dogs, your horses to be hunting wild boar, pheasants, pigeons, rabbits, ferrets, all these kind of species that now make up the pests of the UK, but where they're held in these royal hunting grounds. And on these manorial estates, you typically would have had a rabbit warren, a dovecot for your doves, and maybe even like a carp pond, some sort of contained area for your fish as well. So you've got your 3 major medieval food sources there. So when we think of what, again, is the dispersal history of the rock dove, feral pigeon, we've heard a lot of things being said about when they potentially were domesticated. Did the Romans bring them in? Was it the Normans? Was it earlier? The answer is I don't know, and no one really knows. People make conjectures, but there has been no concrete scientific work done yet. It's getting closer, but nothing that confirms even where they originate from. So some people say Turkey, but there isn't any actual science to back that up, other than, I mean, we can see that they are coming from that general region, but is it in North Africa? Is it further south? Is it further north? Is it further east? We're not sure. It's in that zone, but it's not confirmed that it actually is from within Turkey. So moving on to my actual project. So what we're going to be doing is looking, as was said, using radiocarbon dating, ancient DNA, and dietary isotopes. And so the ancient DNA extraction and analysis, we've got 18 pigeons from the Roman to post-medieval period in Oxfordshire and the UK. And on the left, I've included there, I believe that was one of my pigeons from Lincoln College at Oxford, which is just saying it's a juvenile pigeon ulna. I just think it's fun to see the bones. And then here we've got our radiocarbon center. So looking, and this is right here, is the AMS, or the accelerated mass spectrometer. So after a lot of lab work, you then load your tiny little graphite columns in over here. I don't know if you can see my cursor, but I'm pointing at the far right in that image. And then they run through this really massive, complicated system of different pressure changes and ionization. And at the end, it spits out 3 different levels for your different carbon isotopes. So carbon 14 being what we want, but is the least abundant, carbon 13, and carbon 12. I'm doing radiocarbon dating on 10 of the bones, and then also doing dietary isotopic analysis for carbon and nitrogen on those same 10 bones. So here I've just included a little image. It's a little bit basic, but I liked it. It was all purple. These here are my extracted collagen. So that's those blue tubes. So within those is where, through the whole chemistry process with radiocarbon, you then get this kind of, it looks almost like styrofoam packing, those little packing peanuts, but stringy and long, because it's just pure collagen that's been extracted from the collagen, and then is in there. And then I don't know if anyone here has actually done this kind of lab work. I apologize if I just am fully over explaining something. You then load them into these tiny, tiny, tiny little tin capsules, and they are ran through the different mass specs. So for this one, it's going to be ran very differently than for dietary isotopes. And these are all the different standards that you have to have as well to make sure that your values are true and make sense. So in the way that, at least when I was an undergrad, I really had no understanding of just how much work is involved in these kind of things, where I'd read research papers and I'd be like, what? They only did 5 samples. They only did 20 samples. That's so little. So this is just to give you an idea of, for only 18 samples, the timeline of what we're looking at for my own research, and just how long everything takes. So I am so fortunate in that I have the wonderful Alice Dobinson, who is a PhD student at Oxford, currently on an RA-ship. She has done all the lab work for me, because at least at Oxford, they don't allow non-trained personnel to be doing the ancient DNA, because it's just so prone to contamination and things can go wrong. So I had the lovely privilege of sitting and watching her for the week and a half that we were in there doing it. So that occurred in April, and we've only just got back in June our results back. So we're just starting to look at and analyze the DNA that's come back. Radiocarbon and dietary isotope lab work also took about 3 weeks, where I started in May, and I just actually got back this week my dates as well. And within that, all different kinds of training of all the different softwares that you don't assume, like that you don't think of that you'll need. I did not realize just how much coding is involved with working on ancient DNA. So I've had to be taking courses in R and Linux and learning to code in Python and all these different kind of scripts, just so that you can access, because when we're working with ancient DNA, we're only working with a mitochondrial genome, so it's 16,000 base pairs. That kind of data can't be stored on your own laptop. It would just kill your hard drive. So we're having to work on the server, which is the supercomputer here at Oxford. But then in order to access that, you have to be coding to access and make your changes. So I'm still waiting for my isotopic lab results back. So that's where we're here. We're in that end of June, early July, waiting and starting to write. And then continuing to write, and then I'll be submitting in September. So just to give you kind of an idea, I don't know what the range of people here are in your experience, but just to remember how much work is involved in these kind of studies when you're looking at results, especially when lab work's involved. Yeah, and the scope. But to get into the fun stuff, so looking at my specific project, so while we are only looking at the UK, we also have to remember that the anther pigeon is present on literally every single continent except for Antarctica. I was just fortunate enough that Sean Dirty had collected 2,000 pigeon bones from all over the UK. So that's why my project's focused here. And we have selected sites that we can see here that are all located within Oxfordshire, with the exception of these two. There's 2 samples, 1 site, because they're covering 5 historic periods, so that we have the greatest hope of seeing change in the dispersal history over time. So if we're looking in the same region, then we can make more accurate conjectures about what is happening through time when we see these genetic changes. Of course, we didn't have pigeons all from 1 site that went over those 5 historic periods. So this here, so we can just see a little bit more of where we're situated. And then on the far right, as I know a lot of you aren't over here in the UK, this is the area of the UK that we're working with. It's just right over here in this bottom right corner. Um, so here is the site map, the same map we looked at, but now with what the supposed age for the pigeons are. I say supposed as I got my data back. And I think it's okay to share this with you guys. And just have the firm reminder that if any of you are bioarchaeologists and working with small mammals, the utmost importance of dating. So we had two sites that were Roman, supposedly Roman. We have the Whitehall Farm Roman villa, and then the Barton Court Farm had both Roman and Anglo-Saxon bones. Yeah, we were looking at the dates in the radiocarbon lab meeting from Whitehall Farm Roman villa, and I said to the lab manager, I was like, why? Why does that date look so funny? Like, why is the numbers? Because they're uncalibrated when we first look at them. But this one was just the text display was completely different. And she got a funny look on her face and she said, because that one had to use the post 1950 bomb curve on it. So that means that what supposedly is Roman is actually like 60 years old. But because they're bird bones, so they're hollow, they migrate so well through soil stratigraphy and that they look completely homogeneous with the whole rest of the layer. And there's just really no way to tell. I mean, I was handling all these bird bones myself, doing the radiocarbon lab work, and you just taxonomically looking visually, you cannot tell the difference. So it just was a really good reminder of the importance of dating, especially when you're doing ancient DNA work, where it's so important to be concrete just in where in history we're situated when we're making conjectures about genetic history and dispersal and whatnot. So I don't have the calibrated dates, otherwise I would share them with you, but just that nice little reminder of the importance of generating direct dates for your samples. For any study, really, even if you're doing dietary isotopes and whatnot. So this is the supposed age range. We'll see when I get them calibrated. A lot of them still look good, but the Roman ones, there's some true Roman ones, but there's some very modern fake Roman ones. And then here we have, so where I mentioned earlier with Hernandez Alonso with his work, we've got all of his museum data just mapped over here so we can see the distribution and range. So this is a mixture of both anther pigeons or feral pigeons and fancy domestic breeds as well. So just mapped all over, showing that distribution and range. And then the lovely Sean Doherty, he's done some isotopic work on those pigeon bones that he's got. So here we can see, just so we can kind of get an idea of what kind of questions can be answered. So if you recognize here, there's Einsam Abbey. Several of my pigeon bones come from there. You can see that there's a clear dietary difference. So along where they're concentrated, with the modern feral pigeons plotting much higher than the rest of these older, more ancient pigeons. So using dietary isotopes just to see change in diet through time, where it would be argued that potentially if you're seeing a lot of difference, or if you're plotting them, say, against chickens or people, if you see similar diets between the pigeons and the people, the inference can be made is that they're eating the same things, so living a lot more integrated amongst them, or just seeing if there's change in diets through time can be an interesting component, especially when you're doing radiocarbon lab work. Those dietary isotopes are just within reach, within a little bit more work to have more data, and the ethics of making sure we're generating all the potential answers, because at the end of the day, it is destructive science. Several of my bones, there's just nothing left after they've had radiocarbon dating, ancient DNA, and isotopes. So making sure that we're getting as much possible data out of them, just to justify the fact that it is a destructive science. Yeah. So I'm coming to the conclusion. Hopefully I haven't left anything too horrifically out, but there's just some big thank yous to be had. So my primary supervisor is the lovely Gregor Larson. He's the director of the Paleo Barn, which is the ancient DNA lab that I work through. Alice Dobinson, she is just, I feel she's just holding my hands through the whole process, and making things so much more wonderful. Sean Doherty from the University of Exeter is, whenever I start to fall into that trope of who even cares about pigeons, he just reminds me why they are so epically cool and awesome. And I hope that you guys also think they're kind of cool. And then German is where my modern museum data is coming from. And we're just so grateful for having an open access collaborative network wherein we can share data amongst each other. So thank you guys, and I look forward to some questions.